Well, distinguished guests, ladies and gentlemen, it's really a great honor to be able to participate in this historic event, and I share the enthusiasm of the speakers that this is really going to be a transformative event. And so I want to start off by really congratulating you on the launch of this Technion Integrated Cancer Center. And again, I think I just want to dwell briefly on the notion of integration, because I think we are on a frontier where close integration of basic cancer research, engineering, computational sciences, and clinical research is really going to enable breakthroughs in cancer therapy. And so I think this is exactly the right philosophy, and that you're building a new cancer center from the ground up. And I can say that with enthusiasm because I moved from San Francisco to Lausanne, Switzerland, to be part of another Institute of Technology, the Swiss Federal Institute of Technology, with a similar vision that we're trying to build from the ground up a new integrated cancer center, very much like the one here, and I hope that we'll have fruitful interactions with the TICC. So I believe in this philosophy, and we're trying to make it happen. But I'm not here to talk to you about our cancer center today. I'm here really to provide some perspective into the cancer problem, which has already been alluded to by all of the previous speakers. So the war on cancer, have we won it 45 years on? And obviously, the answer is no, although we've won some battles, and you heard some really spectacular examples of that, including from the fruits of the ubiquitin discovery. But that's unfortunately a rarity, and that's why, of course, we're inaugurating this new cancer center today, because otherwise, if the war on cancer was won, we wouldn't be here. So a premise that I'd like to raise is that, to eventually win the war on cancer, and I'm not as optimistic as Aaron, but I think it's a great strategic goal, but I think we are going to win more battles against particular forms of cancer, but to do so, arguably, we need to better understand the enemy. And I think this is no different than fighting ISIS, that you've got to understand the enemy if you're going to effectively battle them. So I'm just going to, for the general public, I'm just going to give you some general thoughts about, what is cancer? Well, cancer is fundamentally a disease of uncontrolled cell growth and chronic expansion of well-behaved cells in our body, where normal cells begin to proliferate aberrantly, and as they proliferate, they acquire additional capabilities that eventually progress into symptomatic disease, which can be lethal. And so cancer originates from alterations in the DNA code of our chromosomes, mutations in genes. And these cancer cells or tumor cells have altered programs that cause them to stop performing their duties as normal members of the society of cells and organs of our bodies. Instead, they become outlaw cells that are reprogrammed for proliferative expansion. Cancer arises in dozens of organs and cell types in the body. There are multiple subtypes of organ-specific cancers. There's hundreds of genetic aberrations that can drive tumor growth. There's dizzying genomic disarray and histological variety in what we see in cancer. There's remarkable diversity in severity and pathogenic consequences for the affected individual. And there's disconcertingly transitory therapeutic benefits for many forms of human cancer, including for these new generations of very expensive, very sexy, very exciting targeted therapies. In many cases, the benefits are remarkable, but transitory, and I'll come back to that. So in sum, the problem we face is that this is one class of diseases, but it's just an extraordinary, one of extraordinary complexity at all levels: genetic, histological, pathological, prognostic, therapeutic. And so how can we rationalize this complexity? I mean, it's hard to think about all this complexity, and how do you make any sense of it? And are there common principles underlying this daunting diversity? And in a series of conversations, Bob Weinberg and I, who's a professor at MIT, developed a line of thinking which was that the disparate cancers share fundamental qualities, and that this daunting complexity merely reflects different solutions to the same challenge, which is that cancer cells must surmount multiple barriers and roadblocks that are used by the organism to prevent expansive cell proliferation. barriers which can differ from organ to organ, and therefore the solutions are different. And this line of discussion led us to pose in 2000 and reiterated in 2011 the notion that cancer could be in some way rationalized, this complexity, by the acquisition of a series of discrete hallmarks of cancer. And so this was a hypothesis for rationalizing the complexity of cancer, and it's had some resonance, which I guess is why I'm still here talking to you about it today. And so I'm just going to walk you quickly through the notions of the hallmarks, that there are 8 acquired functional capabilities that allow tumors to do something active, to do things that normal cells should not, and typically to do them chronically rather than during the carefully orchestrated activities of cells and organs in the body. And the first hallmark is sustaining proliferative signaling. And this, of course, is the most obvious one because cancer is a disease of expansive proliferation in the number of cells resulting in dispersed or tumorous masses. And so really the notion here, the accelerator is on full speed ahead. We have signals to instruct cells to grow and divide that are present chronically, rather than the transitory signals that are present in normal tissues, which divide in a very stereotypical fashion and then cease dividing. The second hallmark reflects the other side of that coin, which is there are growth suppressors, which actually serve as brakes to prevent cell proliferation, and invariably we see these brakes or tumor suppressor genes have failed and that signals to stop are disabled. A third hallmark, so these are all somewhat quasi-distinct from each other, as I think you'll appreciate. A third one is resisting cell death, because it turns out, Nobel Prize-winning discovery, that cells have an inborn capability to undergo what you might call assisted suicide, and in particular, cancer cells can commit suicide, but the ones that we see in symptomatic disease abrogate this protective mechanism that normally eliminates aberrant cells for the benefit of the organism. So again, another barrier, a protective mechanism of inducing cell suicide. The fourth hallmark is another Nobel Prize-winning discovery of enabling replicative immortality, and that we now understand that there's a counting mechanism on the ends of the linear chromosomes that clock every time a cell divides and a chromosome is replicated, and this counting mechanism will actually shut off cell division when a preset limit is reached. And cancer cells actually abrogate or short-circuit this counting mechanism by a variety of mechanisms, most generally through the induction of an enzyme expressed in stem cells called telomerase, which protects the ends of the chromosomes. So cancer cells enable replicative immortality by disrupting this counting mechanism. The fifth hallmark takes us outside of the cancer cell into the microenvironment of the tumor, and that nests of cells cannot grow without oxygen and nutrients like glucose to fuel their proliferation. And the fifth hallmark is the induction of new blood vessel growth, the process of angiogenesis, which feeds and nurtures the growing mass of cancer cells. There's another way that cells can survive, a very insidious way, and that's the sixth hallmark of activating invasion and metastasis. And this is, of course, the hallmark which causes the symptomatic and lethal disease, where cancer cells become guerrillas that grow by migrating and invading into normal organs, locally and throughout the body, living off the fertile land and disrupting the function of normal tissues and organs. And so this is another way which cancer cells can expand and survive. The seventh has really been has come into focus in the last decade as a hugely exciting area, the notion of deregulating cellular energetics and metabolism, which is really that cancer cells become dynamic and flexible in the way they use nutrients to fuel their growth. So you can consider them hybrid engines. They use alternative energy sources and alternative way to generate energy from different molecules such as glucose and glutamine, and really to restructure the metabolism of cells to provide fuel and biomaterials for cell growth. Because again, normal cells sitting in a tissue are not proliferating, or if they do so, they proliferate briefly. Cancer cells need fuel and biomaterials for chronic cell proliferation. And the eighth hallmark is avoiding immune destruction, and that what we now appreciate is that in many cases the immune system detects a problem and seeks to kill cancer cells, but this attack is abrogated in what we see in symptomatic cancers, that they figure out ways to evade or avoid immune destruction. And again, this is an area now where we have exciting progress in learning how to disrupt these mechanisms for evading immune destruction. So the hypothesis then is that the acquisition of these hallmark capabilities is in general necessary and sufficient to generate a lethal cancer. And that's at least a working hypothesis to think about some of this complexity. But then how are these hallmark capabilities acquired? And we argue that there's two enabling characteristics, of which the first has been known for decades, and that's genomic instability and mutation. And that cancer cells lose the integrity that maintains our genome, that as a fertilized egg instructs the development of an organism as complex as us by really maintaining the integrity of the program encoded in the DNA. But cancer cells just show failures of crucial teams of proteins that protect the DNA of the genome from being corrupted, rearranged, and damaged. And the result is mutations that convey on cancer cells hallmark capabilities. And as was alluded, we now have the capability to sequence very economically the human genome, and that's revealing in extraordinary depth the degree of genomic instability and mutation that's happening in many forms of human cancer. The second enabling characteristic is one that was less obvious, but has come into focus in the last decade or so, and that's tumor-promoting inflammation. And the realization that even though we think of the immune system as, or we hope the immune system is trying to kill tumors, it turns out that there's a dichotomous relationship between the immune system and tumors. And that is this notion that tumors are wounds that don't heal, and that there are cells of the innate immune system that have been designed to fight infections and heal wounds, and they misdiagnose the problem of cancer, and immune cells that normally participate in wound healing inadvertently help cancer cells acquire hallmark capabilities and become more aggressive. So the immune system can help us, but it can also hurt us, and indeed these inflammatory cells can provide many of these hallmark capabilities. And that leads us into another line of thinking that although the cancer cells contain the mutations that are driving the disease, those mutations in genes that drive cell proliferation and other hallmark capabilities, tumors are not bags of cancer cells, nor are cancer cells growing in a petri dish. There's really a complex microenvironment that's created that involve a number of corrupted and recruited normal cells. And so I just want to walk you quickly through those. And the bottom line is that tumors are composed of an assemblage of cell types that communicate and collaborate. And of course, the foundation of this are the cancer cells, and there are now, we appreciate many, this instability of the cancer cells is producing dramatic heterogeneity in many forms of tumors, just illustrated here with the notion of cancer cells that are invasive and others that are not. But there's incredible complexity now, we realize, in differing regions, even in the same tumor. And in addition, there's another exciting dimension, and that is the realization that there are so-called cancer stem cells. These are cells that are more similar to the stem cells, the normal stem cells in the body that help preserve the homeostasis and the integrity of organs, that cells that have turned on stem-like properties which help them to survive. And so there's a whole series of dimensions now of the cancer cells themselves. But in addition to the cancer cells, there are these recruited and corrupted and reprogrammed normal cells. And two of those are the endothelial cells and periocytes, which are induced to form the tumor vasculature, which feeds the cancer cells. There are addition cancer-associated fibroblasts, which are recruited from their normal support functions for many organs and come to populate tumors, along with the aforementioned tumor-promoting inflammatory cells. So these are innate immune cells that normally participate in wound healing and other activities, but instead are contributing to the tumor. And so research across the spectrum from many labs over the last 10 or 15 years has really established that these so-called stromal cells that are not mutant cancer cells are very, very important for the formation of the disease, and now increasingly we realize that they're important for the resistance to therapy. And so functional studies that have been done have really documented the realization that of these 8 hallmark capabilities, these recruited angiogenic vascular cells and cancer-associated fibroblasts and tumor-promoting inflammatory cells can contribute to all of these hallmark capabilities. They can contribute to proliferation, angiogenesis, invasion, resisting cell death. Actually, there are members of these cells that actually inhibit the cytotoxic T cells that are trying to come in and kill tumors. So these cells are definitely part of the problem. And so the main point here is simply that although we're all thinking about therapies targeting the cancer cell, the armed forces of the enemy is complex, and these other cells are really important for the initial development of the disease, as well as increasingly we appreciate the responses to therapy. So that's the second point. We've got these hallmark capabilities and the characteristics that provide them, and the notion that this is an outlaw organ that is really produced, and that is something we have to understand about this enemy. So then, and another thing that's implicit in all of this rearrangement is that cancer involves reprogramming of the integrated signaling circuits in cancer cells and also in these recruited normal cells to convey hallmark capabilities. And so what we appreciate, very exciting time now, is that the cells of our body have integrated circuits, much like the complex integrated circuits that we can create. And so there are many circuits within the body, within a cell that perform differing functions, and I'm not going to dwell on them today, except to say that what we now appreciate is that through genetic mutation and also epigenetic rewiring, cellular regulatory circuits are corrupted to convey hallmark capabilities. And so this is a very exciting realization that we can now chart these aberrant signaling circuits and understand how to interfere with them. And certainly a number of talks at this conference will dwell on that. So those are the notions that we put forward in trying to rationalize this vast complexity. And so I think arguably that this concept is helping to rationalize the wealth of new mechanistic data forthcoming from the cancer research community, because this has been a rather resonant concept now for over 15 years. But an interesting question, which I just want to end with, is whether this concept is applicable to the goal of more effectively treating human cancers. And interestingly enough, we now have smart bombs, or so-called targeted therapies, for all of the 8 hallmarks, as well as the 2 enabling characteristics of genome instability and tumor-promoting inflammation. So we have, and these are just illustrations here, the names are not that important, but the key point is that through the development of the whole academic and pharmaceutical industry studying cancer over the last decades, we have an increasing armamentarium of drugs that target different of these hallmark capabilities. And so that seems, is a very interesting development, and one which you would think might really be helping us above and beyond the traditional approaches of using nonspecific chemotherapy or radiotherapy to treat tumors. And that was the great expectation when we started bringing some of these drugs, such as some of the first ones were EGF receptor inhibitors that targeted the proliferative signaling hallmark. But the reality is that most of the time, these smart bombs are smart, but they're not enduring. And I'll just give you one example of that. So melanoma is an often deadly form of skin cancer, and it becomes deadly when it metastasizes throughout the body. So there's a radiological image and a different one of the liver here. So it's metastatic melanoma has been largely incurable. A drug that was developed that targets the proliferative signaling hallmark and through the mutant genes that were identified, and it has just an amazing response. It melts away metastatic disease. In this radiological image, these are normal tissues here, but this is all cancer. So these are sort of normal. And so this, in 2 weeks, this drug melts away metastatic disease. So this is what we would dream about for a targeted therapy. And that's the good news. The sobering news is that 6 months later, metastatic disease is back with a vengeance. And so this is both the promise and the challenge that we face, that basically most of the time, these drugs targeting individual hallmarks are producing dramatic responses, but they're not enduring because the cancer cells are adaptable. It's a classic Darwinian evolution. 99% of the cells die, but in that population, because of the instability of the genome and the ability to reprogram themselves, variant cells emerge that are resistant. And this is a very exciting and important area of cancer research now. So the question is, so this is in this example here, for example, this quite expensive drug combination of BRAF and MEK inhibitors produces this wonderful response, but most of the time for most patients it's not enduring. So what is the path forward? Well, I think that the path forward, you could hope actually, well, let's target all of the hallmarks, and maybe then it would be impossible for a tumor to adapt simultaneously to all of this, which is a nice fantasy, but it's almost certainly unrealistic due to toxicity. But so the thought that I would like to pose is that that we face a future where perhaps by using rational combinations of targeting differing hallmark capabilities, we can put the tumor in a situation where it really can't efficiently adapt to all of these challenges. And so the notion is you take rational combinations, for example, of targeting angiogenesis, metastasis, and metabolism, or you activate the adaptive immune system and suppress the tumor-promoting inflammation. So there's a number of strategies that one can think about. But even then, they may develop adaptive resistance, but also with new imaging tools and capabilities for monitoring disease progression in the blood and by non-invasive imaging, one can imagine a futuristic battle plan for the war on cancer where we use complex sequencing regimens. And by the analogy to conventional warfare is we don't stand up in lines on the field and march at each other anymore. I mean, you attack by land, by air, and by sea. And so maybe if we imagine sequences, you target immune evasion, but if you see relapse and start detecting it early, you switch to target the oncogenic drivers, and then you switch to target angiogenesis and metastasis, again, in such a way that perhaps the tumor is not going to be able to respond, and we can produce a condition of enduring response. And so this is sort of a hypothesis for the future, but I think that the world of cancer therapy is certainly into one of combinations now, and this is just a way of thinking about mechanism-based combinations organized around these hallmark capabilities. This is a hypothesis. It may or may not prove useful. So again, to end then, I think the war on cancer, the enemy is powerful and diverse in its manifestations, but we're on a frontier where knowledge about mechanisms of the disease, in my mind, is going to produce exciting progress in the battlefields of cancer. And I think that the TICC is going to be part of that solution. And so I'll just end and just mention, the Hallmarks concept was developed by Bob Weinberg and myself, we've had a wonderful collaboration over the decades. And I'll stop there. Thank you.